All right, guys, we are back with week 14. We'll be talking about chapter 8. This will contain pieces from chapter 8 and also the endocrine chapter in your textbook. But chapter 8 is probably one of the least important chapters in the entire textbook. So we're not going to go through each and every slide here because much of it is actually irrelevant to what we're doing in kinesiology. So we'll make this one a bit shorter and we'll start adding a little more complexity behind it in next week's lecture. So be kind of ready for that. And also, your final exam is rapidly approaching, and it's going to be very similar to what you guys did with exam 3, except the content's going to change a little more. And I'm going to ask you guys to look a bit more into some of the molecular and cellular components of what we're going to talk about in this chapter. Specifically, I'm telling you right now what direction we're going to go. Is I want you to start thinking about micronutrients and how they're metabolized and how we utilize them. And you can kind of choose what direction you want to take this in. So if you want to look at micronutrients as far as vitamins and minerals, more specifically vitamins, what role they play in potentially generating energy, such as your vitamin B complexes. If you guys are focusing on kinesiology, you should be well aware of the role and the function and the availability of vitamin B complexes. C, D, E, and K, you should know the differences between fat-soluble vitamins and water-soluble vitamins and what they actually do. But most importantly, you should understand how they're metabolized. And I will give you guys specific examples of what I expect as we get further into this lecture. And then next lecture, we'll talk a little more about endocrinology. But I think what I'm going to have you guys do to make this as relevant as possible is have you do your final presentation on nutritional diseases / nutritional performance and the role of vitamin and vitamin metabolism in that realm. That is where I think I'm going because vitamin metabolism is actually very complex, and I would like to kind of have you guys go about that and explore that world. So let's get into it. So what we're going to talk about in the first part of this chapter is environmental conditions and diseases. And this is where we're going to go kind of quickly through it because this part truly doesn't have much relevance to what we're talking about with kinesiology, but it is important to understand how environment can play a role in the development of disease. So there are a number of specific environmental issues that can impede on human health and human wellness. And these issues can include things such as air pollution, toxic exposure, toxic elements or irritants in the air, in the water, in the work and domestic locations. All these things can have a significant impact on your health. And why does that matter? Well, because what we generally tend to see is people who live in impoverished areas are generally surrounded by these type of pollutions and these type of toxic environments. Not only are they fighting the battle on that front and dealing with environmental issues, but they're also dealing with nutritional issues. And we know that when there are nutritional issues that are being dealt with, whether there's poor diets or there's obesity or there's diabetes or there's the development of metabolic syndrome, that's going to give these conditions more leeway to impact somebody's health because we know that diet, exercise, and sedentary activity all kind of open the doorway for other things to get in and to make our lives fairly miserable. So we're going to talk about how these sort of environmental conditions can kind of get into the body, and we can see that obviously air, water, and soil. How does soil impact us? Well, much of the vegetables and the plants that we eat contain toxins and pollutants that can get into the body. So when we either breathe these things in, or we ingest these things, or we just absorb them in our skin, that's how these toxic molecules can enter the body, and then they get absorbed into the bloodstream. As we know, the skin is highly vascularized, as you can see here. There's capillaries and there's veins and venules here. The lungs are highly vascularized. That's how we get oxygen into the body, and we get carbon dioxide out of the body. And then whatever we eat that goes into the GI tract, well, your small intestines is just filled with vascular components and capillaries, and that's what helps you get nutrients across the small intestines and into the bloodstream. So once we get them into our body, they're easily accessible to get into the blood and then to be distributed throughout the body as a whole. And some of these things that we take in, we can metabolize. Some of these things we store, and if I give you an example of things that we store, things like pesticides and herbicides, those things that we get from the soil and we eat over the course of our lives, they get stored in the body. Their concentrations increase, and then at certain periods over the lifespan, they can become problematical. And they can basically cause complications with health, whether that be neurological issues or issues with the brain. Those things are all kind of associated with the bioaccumulation of herbicides and pesticides that we store within our body, and that's what we call that process is bioaccumulation. So human exposure to pollutants. They're contained in the air, the water, and the soil, and these are generally absorbed through these three systems here. Once these things are in the body, they are generally transported through the bloodstream to various organs where they can either be metabolized and not be detrimental and not threaten our health, or they can be activated, and if they become activated, they can become what we call reactive metabolites. And obviously, when we have a reactive metabolite in the body, this is something that's going to cause trauma. So, of course, we can sunlight and radiation is also something that is incredibly harmful to the body. Sunlight and artificial sunlight. So when we're doing things like tanning beds, you guys are familiar with how reactive oxidative species works. Well, tanning beds like to accelerate the generation of free radicals and actually create some pretty nasty free radical generation. And then there's also toxins such as tobacco and drugs, and you guys are well aware of that. So some things, let's look at this graph here. So some things that are potential toxins are toxic right out of the gate. One of the things like methionine, that's an amino acid. Methionine in very, very, very high levels is something that is toxic to the body, to the cells. So out of the box, methionine, this is an amino acid that's found in potatoes, tropical fruits, meats, cheeses, and so on. And it's a major flavoring component in things like fermented soy. When we have high levels of methionine, they can be toxic. So if we have normal levels, they can be a non-toxic metabolite, or if we have high levels, they can turn into reactive metabolites, and they can affect cellular processes and the molecules and enzymes, receptors, and everything within the cell. So when that happens, the cell has to repair itself. And again, if we can't repair itself, we're going to move into something called toxicity. And this is also true for lots of vitamins and lots of minerals. There are too much of a good thing, and this is something we have to think about when we're dealing with what's one of those popular things with students. What is that thing called? 12-hour energy, right? Or something like that. 12-hour energy drink shots, whatever they're called, right? Filled with vitamin B complexes to help give you energy. Well, or is it 4-hour? I think it's 4-hour energy, right? Something like that. Let me do 4. I think that's it. You can see them at the gas stations, right? So some guy just says, hey, I'm going to give these people this massive bolus of 100% of all their vitamin B complexes. And we're just going to say it gives you energy. And we all know very well that vitamin B by itself, nor the vitamin B complexes, give you energy by themselves, but they're a part of a pretty complex process. They're coenzymes, and they help us create energy, but they don't do it by themselves, right? So when we do high levels of vitamin B, well, those are water-soluble vitamins, so we can get rid of them, and our P is a nice fluorescent yellow, right? So we can get rid of those more readily. Whereas A, D, E, and K vitamins that are fat-soluble that we store, we can take in a high concentration of those that can be toxic, which leads to vitamin A toxicity or vitamin D toxicity. And that is when we have something that can become reactive and can become dangerous. Some other examples of toxins could be farming or weed control or pest control when we take those in via these methods here, right? Those can some components of those toxins can be metabolized, others cannot. And once these things are consumed, they can damage the liver and the nervous system. And then they can also generate these metabolites, right? These reactive metabolites, which also are dangerous to the body, and they can break down and they can cause all types of disruption within the cell, as I kind of told you here, right? And so reactive metabolites have been strongly associated with cellular proliferation and cellular splitting and cellular dividing and unregulated cellular growth, right? So these reactive metabolites, they can kind of re-hardwire the cell, program it to do things it shouldn't do, and then one of the major results of that process is not only toxicity, but the development of cancer and things like that. So something I want to kind of pose to you to get you to think about some questions here is, what happens when we're bringing in multiple toxins at one time? Let's say we're living in Los Angeles, right? Beautiful Los Angeles, and we know that that orange cloud is looming above everyone there. And I'll tell you what's in that orange cloud as we kind of get further into this lecture. I'll tell you some of the things in there that cause cancer. Let's say we're breathing in some of these toxins in the air, and we all know the quality of water in Los Angeles is not very good either. And let's say the soil from Central California, where all of our plants, all of the produce is produced, right? The almonds, the avocados, all the vegetables that we eat. And let's say the wind blows through that soil, and that these toxins here, those chemicals that we use, such as pesticides, they become airborne. And now we're breathing in the LA air. We're drinking the water in LA. We're breathing in the pesticides and the herbicides that are being blown about throughout Central California. Well, we have this massive exposure to all these different toxins at one time. So what do you think happens? Well, these things accumulate, and this process here, we develop more of these reactive metabolites. We have more cellular damage. We lose the ability to repair the cells, and then we run into this kind of chronic toxicity that leads to other conditions in the body, such as chronic inflammation. So, and another example of this sort of potential toxin would be something called xenobiotics. These xenobiotics are in drugs, they're in pesticides, they're in cosmetics, they're in flavoring agents that we eat, they're in fragrances, they're in food additives, they're in industrial chemicals, they're in environmental pollutants, they're everywhere. And these substances, when we take them into the body, think about drugs. Think about if you're taking a prescription drug and you're on it for a long period of time. They can be metabolized to either a toxic or a non-toxic metabolite and eliminated from the body. So some drugs, most of them are tested and they become non-toxic metabolites. But if we overuse the drug, right, that's why when they prescribe drugs, they have to consider the dose, and they have to consider your body weight, and they have to consider how long you're going to be on that drug, right? Because if it's in your body for too long of a time, at too high of a concentration, we go from a non-toxic metabolite to something that is a reactive metabolite, and then your body can no longer clear it, and then it begins to damage your body. So, and then again, if the let's say the drug that you're taking, if you're taking it for too long of a period of time, then this is going to be something that's going to cause chronic toxicity, and it's going to impair your cell's ability to repair itself. So those are just a couple examples of how this environmental condition kind of works in the body. But it's not just air, water, and soil, but it's also in medication, right? It's also in food additives. It's also in fragrances, right? So we're truly living in a time where there is a chemical stranglehold on the United States as a civilization, because we rely so much on chemicals and rely so much on drugs and so much on additives, and we rely so much on pesticides, and that we're just taking these things into the body on a massive, massive scale. So let's move away from this slide and talk about some of the differences between indoor and outdoor gases that can potentially be harmful and toxic to the body. Of course, the most popular one that I'm referring to is radon. I'm sure you've all heard of radon before, or you may have tested for radon gases in your house. Basically, radon is it's a colorless, tasteless, and odorless gas that has been associated with the development of pretty aggressive lung cancer. And this gas can reside at really dangerous levels inside of the house. It's also been found in schools and other buildings. And exposure to this gas is the second leading cause of lung cancer in the United States after smoking. So this is a pretty noxious gas that certainly can devastate the lungs and the body. So this gas, it forms naturally in the soil. So you can see a picture here. It's really kind of, let me see if I can fix this, because that does not look good. Let me see if I can make it a bit more clear for us. Yeah, that's a little better. So radon, it comes from the soil. It forms naturally, and it's basically formed from uranium in soil and rocks. And it breaks down to form this thing called radium, which then turns into this gas called radon. And once it's formed in the soil and in the rocks, radon can enter the home through cracks in the walls or basements or the floors of the foundation. And other openings in the house, and essentially it will get trapped in the house, and we breathe this gas in, right? So again, let's go back here. Breathing this gas in is incredibly harmful. And then as it decays, because it gets trapped in the house for a very long period of time, as it decays, it releases these radioactive byproducts that are inhaled, and that's believed to cause the catalyst to be for lung cancer development. And because it's found in rock and soil, it can essentially be found anywhere. And one of the major areas where radon is a constant problem is up here in Canada. They have some pretty high levels of radon poisoning, and the homes there have to constantly be tested for radon. And again, it has nothing to do with how old the home is or what the home is made of. It's just something that's in the soil, and it's something that we have to be aware of. And if you don't have your home tested for radon, you can certainly develop cancers, lung cancer being the most prominent one in radon poisoning. So on this next slide here, just in case you haven't been aware of what radon is, I just kind of have this showing you how high the cases are in Canada. So you can see that these are pretty high levels, and depending on where you are in Canada, some of the levels are higher than others. So this district here, Leeds, is the highest. And let me see if there's any other ones that we recognize here. Ottawa, they're high. Toronto is pretty high. But yeah, Canada seems to have a really tough problem with radon in their soil. So they're constantly getting tested. So let's talk a bit about what toxicology is. So toxicology, you probably heard about chemicals, right? Especially synthetic chemicals, the type that we see in nonstick pans. There actually was a movie about this. I don't know if you guys have watched it or not, called Dark Waters, and it was all about Teflon and their company that was basically putting thousands of pounds of waste product from this, making these Teflon pans, into the rivers, and then the rivers were poisoning everybody in the surrounding area. Everybody was developing cancer. So we have these toxic chemicals. Again, let me just kind of go back to let you to kind of reinforce this idea that we can get these. Where's that slide at? Maybe I didn't have it on the slide. I just said it. I said before that we can get these toxins, right, from pesticides and drugs and cosmetics and flavorings and fragrance and food additives and things like that. So some other sources of this of toxins would be these nonstick pans, right? So you've heard about synthetic chemicals that they use in nonstick pans. They use the same synthetic chemicals in cosmetics and even aluminum cans, right? So we're taking in these toxins here, these synthetic chemicals here. And if you're a lady, you're putting it on your face here, and we're also getting it here. You're making your eggs or whatever you're using this nonstick pan for. There's all these products on there. And all of these toxins that are found in these products, they disrupt hormones. They disrupt hormone production, hormone signaling, hormone transient effects of hormones in the blood. So again, when we have this bioaccumulation because of the exposure we have to all these chemicals, we run into problems. And of course, anything dealing with hormones, where we have an increase in hormone production and signaling, generally leads to messages that tells cells to divide and to get bigger and to grow. So there's also BPA. I know you guys are familiar with that in a lot of the plastics. That's another chemical that is found to disrupt hormone signaling, hormone reception, hormone sensitivity with receptors on certain cell lipid bilayers. And in laboratories, things like BPA and synthetic chemicals used for nonstick pans and in aluminum cans, they have been shown to also increase the release of molecules called interleukin-6 and interleukin-8. And you guys are familiar with what those are. So not only are they potentially carcinogenic, they're pro-inflammatory, right? So this toxicology generally includes studies of physical agents such as radiation and heat, but it also includes the study of chemicals on human health. And this was profound. I found this and I wanted to show this to you. Roughly 70 to 100,000 pounds of chemicals, including known carcinogenics, are produced each year in the United States. And look at this, of all the chemicals that are allowed to be produced in the United States, roughly 1% have been tested to determine how they impact human health. So here is a perfect example of how profit over health seems to be the main concern in this country, because our government allows these things to be created. Not only do they allow them to be created, they regulate the creation and the disposal of some of these products, and these chemicals enter our body and they cause havoc. Sorry, I've been talking all day today. They create these reactive metabolites, and then these things disrupt everything within the cell. And again, if they disrupt the cell, they disrupt the tissue. If they disrupt the tissue, then we're going to have systemic issues as a result of that. So that was a very interesting statistic that I found while I was doing research on this particular lecture. So let's move on. So air pollution, right? So now we're talking about outdoor. When we were talking about radon, we were talking about indoor. Now we're talking about outdoor. This is another one that there's airborne microorganisms, right, that have long been a major cause of mortality and morbidity here in the United States and across the world. There are hazards that are in the air, and we'll talk a little bit about Los Angeles here. So when we're talking about outdoor air population, Obviously, these things are going to have a tremendous impact on human health, and they're primarily caused by industrialization and contamination by industrialization. So all of those wonderful smokestacks that we see on the skylines of certain cities that are industrialized, that are just spewing these irritants and these microorganisms into the air that we are bringing in. Again, so if any of you are from the Los Angeles area, you've noticed that smog that's just kind of looming about the city. Well, that smog contains roughly 6 primary pollutants, which are sulfur dioxide, ozone, nitrogen dioxide, lead, and then there's another thing called particulate matter. And these things are all making that wonderful orange cloud that just looms above the city. And these things, especially ozone, is highly, highly reactive to the lung. And all these things, all the things that I mentioned, their particle sizes are all, they're different sizes, but most of those gases that I had basically just mentioned to you are capable of getting into the bronchi and the alveoli and basically destroying the tissue here. So they scar the lung. And when you have constant exposure to those pollutants and they're getting into the lung, especially ozone, that's the nasty one, they're going to cause damage. They're going to cause things like COPD. They're going to cause asthma. So lots of issues there. So let's talk a little bit about ozone, because there are 2 types of ozone, right? We have the friendly ozone, right? We have the one that is the good ozone, which is in the stratosphere, right? And we know that it's basically produced by the interaction of ultraviolet light, radiation, and oxygen, right? And we know that it is a protective layer. It promotes life on Earth, and it absorbs most dangerous UV radiation emitted by the sun so that the sun doesn't turn us into crispy critters. So we call that the good ozone, right? So, and that's represented by O3 here. Now, when we talk about the bad ozone, which we have over here, this ozone is formed by the reaction of nitrogen oxides and organic compounds in the presence of sunlight. So this thing here, we get this kind of concoction where we make this harmful ozone underneath the stratosphere, where we basically bring it in. And you can see here that the heat, the sunlight, and the pollution, they all kind of make this slurry to make this O3, which is essentially a type of radical, right? It's a type of free radical. And these are molecules that we breathe in, and they cause significant damage. So they produce a type of free radical, which is going to injure epithelial cells along the respiratory tract and in the alveoli and in the cells within the lungs. And then not only do they cause destruction by themselves, but they also release inflammatory mediators such as the interleukins, right? So if we're constantly breathing it in, we're constantly damaging our tissue, then we're going to have a constant chronic inflammatory response. And of course, when we have inflammation that's on all the time, that is one of the catalysts to developing certain types of cancer. So it's truly a horrible concoction. And when we live in places like Los Angeles or San Francisco, you're going to be more susceptible to something like that versus where we're living here in Northern California. We're going to have way less interaction with that. So for those of you that have grown up in this area and you have a cell phone, you would remember that when I lived in LA, we would always get messages on our phone telling us what the air quality is. And the reason they would tell us what the air quality is because if we're outside and we're breathing in those pollutants all day long, well, they're going to have substantial impact on our lungs and on our physiology. And if we are doing that throughout the summer and throughout the year, well, you can imagine the accumulated effect that is going to have on our lungs, right? And not only that, but also systemic inflammation. So of course, our lungs, we have ways of dealing with these agents. We develop mucus, which basically helps protect, right? The mucus that surrounds the alveoli protects the sacs. It acts as a second membrane, which basically acts as a filter. We have the cough reflex, right? So when things come into the alveolus or the bronchial tubes, we can cough them out. However, if it's too small and we can't remove them, that's when the scarring begins to happen, the chronic inflammation, and unfortunately the lungs will bear the brunt of the adverse consequences. And one of those consequences are emphysema. So you guys are probably familiar with that. Emphysema is not just a condition that is surrounded by, or not just a condition induced by smoking, but it is a lung condition that causes shortness of breath, right? But generally we say, okay, well, emphysema is caused by smoking. It absolutely is, but also pollution, both indoor and outdoor, can cause emphysema. And basically what happens is the air sacs in the lungs, they become damaged, right? So we go back here. So let's say the mucus just isn't doing its job, and these individual sacs that basically expand and contract every time we breathe in, they become damaged and scarred, and with that we lose function of those sacs, and this creates larger air spaces instead of small ones, right? So what begins to happen is, let's just draw some pictures here. In healthy alveoli, we have small little holes like this that basically allow air to get into those air sacs. When we begin to develop things like emphysema, those sacs, those holes in the sac get bigger and bigger and bigger, right? So they go from something like this, which is healthy, and these are all within these little sacs here, and they go into something absolutely way larger than they first were. So we create these larger air spaces, and this reduces the surface area of the lung, and in turn, it also, the amount of oxygen that reaches the bloodstream is impacted. So this is essentially a scarring condition that occurs, and over time, this scarring reduces lung capacity. And if you look at this individual here, he's very withered and skinny. The breathing has become very difficult, and there's a lot of energy and a lot of calories that are dedicated to simply breathing. So this type of condition will ramp up your metabolism quite a bit, because breathing is such a laborious task, okay? And now, as long as he is relatively inactive, this person can maintain O2 saturation. But the simplest tasks, such as walking up a flight of stairs, will alter his ability to basically take in oxygen and get rid of CO2. And basically his CO2 will become very high. It will stay within the body, and that will be his demise. So that is basically how emphysema will impact the body. And again, emphysema is a combination of ozone, right? A combination of some of that sulfur dioxide and all that other nasty stuff that is actually in the air. And to give you a clearer picture on what emphysema looks like when we see the changes in the lung, you can see here that these little holes, right, are very tight and very secure. But when we get something like emphysema, you can see that those holes get much, much larger. So this would be a lung that has been damaged over a very long time by either cigarette smoke or pollutants or a combination of both. Another one is asthma. You guys are very familiar with asthma. Asthma can be caused from cigarette smoking, secondhand smoke, right? It's an air pollutants can cause asthma. And we know that asthma happens in children with things like pollen, or things like food or drugs, or dandruff from cats, dandruff from cockroaches, of all things. They can shed, and they can cause an asthmatic reaction. And we know that an asthmatic reaction is essentially an inflammatory response, right? So an inflammatory response will occur because there is some sort of irritant that has gotten into the lungs, and it's causing inflammation. So basically this inflammation, these individuals become hypersensitive to these irritants, and they will basically have an extreme inflammatory response due to that hypersensitivity. So it basically goes overboard. And this is an immediate reaction, and this will secrete all types of inflammatory mediators. It will release antibodies into the blood, and then this will result in, you guys should know, mast cell degranulation. We've talked about mast cells in the past. Then we will have the release of histamine, and then we'll have other inflammatory mediators that will come to the rescue just based upon this pollutant, right, that was brought into the lungs from the air, right? So that's slightly different than the emphysema. This is more of an allergic reaction, where this is damage here that's caused for a long period of time. So then, just to kind of give you one more flavor of another type of toxin that we are living with, would be lead, right? So you guys are familiar with how detrimental lead can be to the body, and we know that lead is essentially a metal, and we are exposed to lead due to industrialization, so industrial exposure. Things like smelting of metal and lead-based paints, these things basically introduce lead into our living spaces. And these can be, the damage can be very substantial with lead poisoning. And a lot of the lead exposure kind of depends on where you're living in the world. So some countries are far more industrial than we are, and they don't have the regulation that we do. So there's way more exposure to lead in some of those places than here in the United States. But the United States is rapidly trying to remove lead wherever they can. Again, schools and paints and things like that. Lead seems to impact children much more than it does adults. And this is due to how rapidly children are growing. So think about the development of kids versus development of adults. So lead seems to impact the brain and bone marrow, and it causes anemia, and it seems to be far more detrimental to children. They are making more, so children, one of the reasons it impacts the brain is because children are making more rapid growths of the brain, right? So they're increasing dendritic connections in the brain, they're increasing brain size, they're developing particular components of the brain. So when lead is introduced to stages of development in human physiology, it's very, very dangerous and toxic. And also, this is the lead is readily absorbable, and it binds to, of all things, protein. So what makes lead accumulate in the body is that it has the capacity to bind to disulfide bonds, right? You guys know what disulfide bonds are. These are when we have a protein that is made. No, let me draw this. When we have a protein, let's see, draw. We have a protein being made, right? And we have these amino acids bound together. And then we have the protein becomes folded, right? Let me find, where's that, what happened to my drawing piece? Here it is, shapes. When the protein begins to fold upon itself and it makes these tertiary structures, right? The reason the protein folds upon itself is because of the bonds that are formed between amino acids. And what happens is lead is capable, let me find this one more time, lead is capable to binding, let's change the color. Let's do, lead sounds like a, no, that's the wrong one. That sounds, let's do black. What happens is that lead will bind to places where bonds are occurring. And the last thing I wanted to talk about with lead is that lead has also been associated with changes in calcium metabolism. So if we have these pretty abrupt changes in calcium metabolism, well, that's going to have a major impact on skeletal muscle function, neurological function, gastrointestinal function, and renal function. So it's been associated with causing toxicity in those tissues as well because of how it interferes with lead. So let's move on here. So what I wanted to show you on this slide before we get too far out of the lead discussion here is I wanted to show you that these disulfide bonds here that connect proteins together, right? You can see this strand of amino acids here, and then it kind of comes around here, and then we have this disulfide bond that is pulling those two strands together. Well, that is where Mr. Lead will bind to, and then we did on the last one, it was black, so let's keep it black. So again, the more disulfide bonds that we have, the more lead is going to bind to those tissues. So I just wanted to show you that. And then here's another thing that was kind of better than my drawing. I told you that it's also lead also interacts with calcium and calcium binding. So here you can see that lead can bind to some of the same spots where calcium is supposed to bind. So lead can interfere with calcium metabolism. Here lead is black, here calcium is green, and you can see that they bind to the same domains as calcium. So this can impact calcium function, whether calcium is active or inactive, because we do have a lot of inactivated calcium in our body, and this can also impact secondary messengers and cellular signaling. So that's all I wanted to kind of talk about with that. So now that we are done with this environmental thing, I just needed to talk about it just to kind of give us a different flavor of science here. We are now going to get into nutritional diseases. And this is where I'm going to task you with what your final assignment is going to be. So when we talk about nutritional diseases, and I'm sure you've seen this in nutrition classes that you've taken, we could be talking about too little nutrients. So this dietary insufficiency would be basically malnutrition, right? So this is basically meaning that one or more of the following components are missing from the diet, right? And those components would be carbohydrates, fats, and proteins. And we're not getting sufficient energy for the body to basically give ourselves calories for daily needs, right? They're not being met, and we're not bringing in enough of those things. They could also be just amino acids and fatty acids. These, of course, are the building blocks for synthesis of proteins and lipids. So if we have any sort of dietary insufficiency where we're not getting enough amino acids and fatty acids, that can lead to too little nutrients, which would have an impact on the building components of our body. Vitamins and minerals, this is what I want you to pay attention to, which we all know function as enzymes and coenzymes. They do not produce energy by themselves, but they are a part of the process. These are vital for metabolic pathways, and they're important for also structural components, right? So we could get too little of them, which could cause serious complications. And then we can also have something that's called secondary malnutrition, which results as from a consequence of malabsorption of nutrients. So we could have impairments of utilization or storage or loss of our ability to uptake some nutrients into the body and use them. But another one could be excess amount of calories. So we could have obesity on the other end, right, which is right here. So we could have too little nutrients. We could have isolated deficiencies, which is what I talked about as that secondary malnutrition, which is just we have like the inability to absorb things for whatever reason, and they do exist. And then when we talk about this too much nutrition, we're talking about dietary issues where we are basically developing obesity and type 2 diabetes, and we're also developing metabolic syndrome and all the other comorbidities that come along with that. So let's just look at an example of what a condition looks like when we have a primary malnutrition, and we're just not getting the right nutrients. So on this slide, we see a condition called kwashiorkor. This is a condition that occurs when protein deprivation is relatively greater than the reduction in total calories. So what does that mean? So that means that not only are we getting very few calories, but we're also getting protein deprivation that is more impactful than the calories themselves. So this is to say that when we have protein deprivation that is relatively high and more severe than a deficiency in total calories. And this is commonly seen in children who have been weaned off breast milk too soon and have been fed almost entirely carbohydrates. And one of the things that happen here is that edema occurs, and you can see that in the stomach here. We have issues with the central nervous system. We have changes in the skin and the hair, and we get fatty liver resulting from reduced synthesis of lipoproteins. And we know that lipoproteins occur in the liver, and they help us develop transport vesicles for cholesterol. So this condition, again, is when children are removed from the breast too soon, and of course milk has high levels of protein in it, and then that protein that is necessary for growth at such a young age is replaced with carbohydrates. And to go back to our lesson that we talked about with hemodynamics, we should understand that the edema is going to occur because the protein that is circulating in the blood is going to decrease. So that's going to decrease or change onconic pressure. So when we talked about the two types of pressure in the capillaries, I told you that the onconic pressure remains pretty consistent. And if there's any changes in that onconic pressure, that's going to change how movement of fluids out of the capillaries and then back into the capillaries are going to change. So if we ultimately change the amount of protein in the blood and we're producing less proteins because the liver is impacted, then that's going to change onconic pressure, which is then going to change how we transfer fluids across the capillary. And you can see again one more time that we have a bit of edema there. So on this slide here, we have another example of deficiency in nutrition. And this condition is called marasmus. And this one here has nothing really to do with protein, but it's overall lack of calories. We can see that children that suffer from this overall lack of calories, there's this stunting of the growth. They're usually 60 to 70% below average weight of children their own age. There's a loss of subcutaneous fat, and you can see how emaciated this child is. There's muscle atrophy as well. So generally, the extremities are emaciated. They have multiple vitamin deficiencies, and they have immune deficiencies. And one of the major immune deficiencies that occur in this condition is loss of T-cell mediated immunity. So their adaptive immune system is also jeopardized. So the previous one that we talked about here, this one is primarily a protein deficiency, where this one is a deficiency in all macronutrients. And interestingly, look at how edema plays a role when there's changes in protein concentrations in the bloodstream, which will produce this swelling, versus the lack of calories. So it looks like two completely different types of bodies. So now, on this slide here, we have an example of the opposite end of the spectrum, which is going to be obesity and diabetes. And we know that this happens when we have an excess amount of intake of calories, and we do not match that intake of calories with calorie utilization. So we begin to develop weight, and then with that weight development, we start to develop things like hypertension, and hypertension is super impactful in type 2 diabetes. We know that this will basically impact the 6 miles of capillaries that we have within the human body. We get insulin resistance. Insulin resistance is generally caused by this high serum lipid issue, because we know that one of the main hits that happens with insulin resistance is when we have high levels of free fatty acids. We know that free fatty acids may be the number one culprit in developing insulin resistance. We will develop type 2 diabetes, which basically is an umbrella term that basically includes all of these things here. We will develop atherosclerosis, and we know that this is also associated with high serum lipids. We know that LDL, low density lipoprotein C, is the main culprit that basically initiates lesions in the endothelial cells of the vascular system, and this causes the plaque to develop. We also develop osteoporosis, and osteoporosis is also caused by high levels of insulin when somebody is developing type 2 diabetes. So why am I sharing this information with you? Okay, you know what type 2 diabetes is. You know what vitamin deficiencies are. You know, you've probably seen these two things in nutrition classes before. So the reason I am sharing this with you is I am trying to prepare you for your final exam in this course, and I'm not going to let you out of this course easy. I'm going to pull up something for you guys really quick, and you're going to have to kind of show me what you learned and show me how well you're beginning to understand cellular mechanisms. And this is what I'm going to have you guys exit with in my class. And the stakes are going to be pretty high because this is not going to be easy for you guys. So when we are talking about vitamin deficiencies, like I said earlier, I said you might be asked to do a presentation on vitamin deficiencies. You might be asked to do a presentation on something else. I'm telling you right now that I'm going to ask you to do either a presentation on vitamin deficiencies, or I'm going to ask you to do a presentation on obesity, metabolic syndrome, type 2 diabetes, and any part of that you want to talk about. But I am going to ask you to focus on mechanisms. So what is a mechanism? Well, let's look at vitamin D. Here we go. This is vitamin D. For vitamin D to be taken into the body and to be utilized, it's a pretty complicated process, right? We need to have dehydrocholesterol. We need to have pre-D3. When we get sunlight, that pre-D3 gets converted into vitamin D3 in the skin, and then that will be converted in the bloodstream to a usable source of vitamin D, but that vitamin D has to go into the liver, and then it has to go into the kidney, and it's a very, very complicated process, and this is what I'm going to ask you to explain to me. Is you can pick a vitamin D, you can pick vitamin B, you can pick vitamin A. You are going to have to explain a deficiency to me, but you're going to have to tell me how the deficiency occurs on a cellular level, not just say, okay, well, this is what vitamin deficiencies do. I need you to show me how the deficiency starts. And this is where you're going to have to go to the research. If you wanted to talk about diabetes, which is going to be one of your other possible topics, right? You are going to have to walk me through a specific part of the pathology. And you and your partner can pick whatever part you want. If you wanted to talk about beta cell failure and tell me how that happens, well, you're going to need to look up something like this. Here is the cellular mechanisms leading to dysfunction in beta cell physiology. So the beta cells, they require calcium signaling, they require glucose to enter the beta cell, and when the glucose enters the beta cell, we have this conversion from ATP to ADP, and basically it's a very complicated process which allows these insulin vesicles to be released into the blood. Insulin doesn't just be released, it's not just released into the blood at any time. It has to be heavily regulated, and one of the major components is glucose coming into the beta cell, because glucose, the beta cell will act as it will basically act as a meter that says, oh, there's this much glucose, let's release this much insulin. But before it can be released, all these other cellular components have to be activated, right? But then when we get into beta cell dysfunction, you can see how these red lines indicate changes in the beta cell, and you can see that free radicals are produced, and the free radicals being produced will destroy the beta cell. So that is an example of cellular mechanisms. So you and your partner will be asked to present on these mechanistic things. And now, is that going to be something with diabetes? Is that going to be something with vitamin deficiencies? It's going to be entirely up to you. If I were to ask you, or if you wanted to present on diabetes and mitochondria dysfunction, right? That's a big one. You might have to present on something like this. Okay, you might have to tell me how nutrient overload, which is fluxes of glucose, lipids, and amino acids coming into the mitochondria, because when we develop diabetes, at first we will have a massive surge in glucose, and then the tissue will become insensitive to insulin, and then glucose can't get into the cell. Then we'll have a massive surge of fatty acids that come into the cell, and they'll get processed in the mitochondria. And then when that stops working, we'll get a massive surge in amino acids that come into the mitochondria, because we'll get into a ketogenic state. And then this will cause, all of these things will cause damage to the mitochondria. And here shows you how the mitochondria undergoes dysfunction. So this is going to be your project for your final exam. Now, the other part of it is I'm going to allow you to talk about exercise. So if you are talking about vitamin deficiencies, let's say we start talking about, let me go back to that vitamin D. Where's it at? We talk about vitamin D, and let's say you're talking about this pathway, and when there's a deficiency, or you're not living in a place where there's sunlight, such as Siberia, right? When you live in Siberia and there's no sun for like 7 months, the children in schools, they have to be exposed to this UV light. So they bring these UV lights into the classroom, and they make the kids sit there so that these pre-vitamin D precursors can get converted into a usable form of vitamin D, right? So you might have to talk about how a vitamin deficiency impacts the body. And there might be a hiccup here. There might be a hiccup here. There might be a hiccup here in the parathyroid gland. That's up to you and your partner to describe. I'm giving you total freedom, but I'm picking things that are complex purposely because I want you to try to talk about this. So to kind of help you guys along the way with this, I'm going to give you an example of what some of my expectations are. And this is going to be a very kind of brief explanation, but I would like to see something like this. Again, either in the realm of diabetes or in the realm of micronutrient deficiency. So we're going to kind of talk about this complex system here of endocrine communication in diabetes and how the endocrine signaling is altered when we have changes in adipose tissue. So this is kind of a representation of a neurohormone relationship, right? We have hormones, we have neurological responses through central processing of this central nervous system. And then the way these two organs communicate is going to impact how the body uses energy or takes in energy, and this could disrupt the energy balance. So this is kind of an example of what I will be looking for when you guys do this final project. So this neural hormone mechanism here that we're going to talk about, this regulates energy balance in the body, and it also regulates body weight. And this relationship here is generally divided into three pieces, which is going to be the peripheral or the afferent system. Then we have the central processing system, and then we have the efferent system. So these are the main components. And something that you might not be aware of is for a long time, adipose tissue was just considered to be this tissue where we store and house fat. But through research, we've realized that adipose tissue is an endocrine tissue. So it secretes hormone messages that will basically communicate with the hypothalamus and other parts of the brain. And the hypothalamus will basically tell the body how to respond to these signals. So when we have the expansion of fat tissue because we're taking in too many calories, then the expansion of this tissue also changes how these signals are sent. So in this main component here, in this afferent system, that has adipose tissue. We have things called leptin. I'm not sure if you guys are aware of what leptin is, but leptin is released from adipose tissue, and this is a hormone that communicates with the brain to tell the brain that the body is satiated, that we have enough food. So when we are normal, healthy individuals, we have a certain amount of leptin that is secreted that tells the brain, I am full, I no longer need to eat, and I am going to stop eating now. So you get this feeling of satiety, and then the hypothalamus says, okay, I got it. And it's basically going to say, okay, let's limit our intake of food. Let's turn it off. Okay. Now, when we have changes in BMI and we have changes in our weight, this signaling also changes. So ultimately, one of the mechanisms of developing obesity could come from changes in leptin. Leptin regulates food intake, therefore it will regulate body mass. It plays a role in pro-inflammatory immune responses. It plays a role in angiogenesis, which would be the development of more capillaries. Lipolysis plays a major role in lipolysis. Lipolysis can be disrupted when leptin is changed and secretion rates are changed. And it's believed to be one of the major mechanisms of obesity. So this would be an example of basically how obesity might be initiated. And this is one of several theories of how obesity is initiated. So this is an example of something you could do. It's like, okay, we want to talk about leptin. And we want to talk about how changes in leptin is going to change the physiology and basically accelerate or exacerbate obesity. So you might talk about, and I'm going to just be very generic here, that it plays a role in regulating body mass via this negative feedback mechanism between adipose tissue and the hypothalamus. So this is going to communicate with this. And how does it do that? Well, the hypothalamus will have, leptin can cross the blood-brain barrier, right? And this is why we talked about receptors in our earlier lectures. So we're bringing it all back. And we know that leptin can bind to these receptors, and this could have a physiological response, which will basically determine how we use energy. And if the receptors become desensitized to leptin because maybe there's an oversecretion of leptin, which happens a lot in diabetes. We get this oversecretion of things. And just like insulin, if it's oversecreted, the insulin receptors become desensitized to the message. So let's say that leptin resistance is characterized by the receptor not recognizing leptin. And if the receptor can't recognize leptin, then the hypothalamus doesn't receive this signal. And then the hypothalamus, which I told you is this central processing area, it can't send out the right information to either be anabolic or catabolic, right? So if it can't shut down the feeling of satiety, well, then we are going to have a lot of food intake, right? And that is going to disrupt energy balance. So this is an example of what I expect from you. I want mechanisms, and I want to have lots of details about these mechanisms. Let's talk about another one. Let's talk about adiponectin. And adiponectin's not on here. It's not represented, but I'm just giving you another example of a mechanism that is associated with diabetes. Adiponectin, if you don't know what it is, it's also produced in the adipose tissue, and this has been called a fat-burning molecule. Adiponectin directs fatty acids to the muscle for oxidation. So if we have changes in adiponectin, let's say we have a decrease in adiponectin, that is going to decrease the directing of fatty acids to skeletal muscle, right? And we're going to have a decrease in oxidation by mitochondria in skeletal muscle. Now, if we have a surge in adiponectin, right? Let's say we lose balance and we have this surge in that hormone. then that means it's going to direct more fatty acids to skeletal muscle to be metabolized to create ATP. Okay, now if we have a surge of adiponectin, well, that can overwhelm the mitochondria, right? And if we have too much fat coming into the mitochondria and we're not exercising, so we're not getting mitochondrial adaptations, that means the mitochondria is going to become overwhelmed with this surge or this flux, that's a mitochondria, of ATP or of fatty acids. And what's going to happen is we're going to start developing these wonderful free radicals, because those fatty acids and those electron carriers, they can't handle all of the electrons coming into the mitochondria. So the mitochondria is going to start to falter and it's going to start to make free radicals. Do you see how this is all coming together now? And if we have changes in leptin or adiponectin, that I told you that is also pro-inflammatory. That's an arrow, which means inflammation is going to go up. And you guys know all about inflammation. So you should be able to tell me, what are these IL interleukins that's an IL-6. I'm still writing with my right hand, so just bear with me. There's IL-6, IL-8, right? IL-6 is specific to skeletal muscle. That's a myokine. So all these things are interacting now, and this is what I expect you to do, is make this comprehensive, right? Draw from what we've talked about earlier in the semester with hemodynamics. Draw from what we talked about early in the semester from receptors and phagocytosis and changes in DNA. One of the things that happens with DNA, especially with leptin is created in one of the obese genes, right? So leptin, there could be change. Let me fix this. There can be changes in DNA which causes changes in leptin secretion, right? This is my double helix. There we go. That looks like DNA. Let me just draw some lines here, right? So maybe you might want to take the genetic road and say, hey, some people are predisposed to type 2 diabetes because of genetic disorders. And if we look at this one gene where leptin is produced, they call this an obese gene, because when this gene is overactivated or it's not regulated, we get some big bellies and we get some obesity, right? So now I'm just tired of talking so much, so I'm just drawing pictures. Okay, that guy's obese. He's happy about it. Okay, so this is what I expect from you. And we can go through all of these. We could talk about, that one's not being erased. Let's get rid of that. We could talk about changes in the pancreatic beta cell, right? Let's say you want to talk about leptin and you want to talk specifically about the receptor and you want to talk about leptin resistance at the receptor level. Well, you're going to have to look into some papers and you're going to have to say, hey, we found how leptin and the leptin receptor activate the signal in the cell, or in this case, you can see how it activates gene activation, right? And you can say, okay, well, in when we get leptin resistance, let's just say that the tyrosine molecule 985, so that means it's the 985th amino acid in this protein, and it's a tyrosine. Let's say the tyrosine is not phosphorylating. You see that P there? That means that it's becoming active. And if this doesn't become active, this can't communicate with this chaperone protein, which is SHP2. And then SHP2 can't activate GRB2, and GRB2 can't activate ERK, which is a gene transcription messenger, and therefore we don't get gene transcription. So there's so many different ways you can take this, and this is what I expect from you. Okay, so I am not going to kill a dead horse here or beat a dead horse. You guys understand what I'm looking for. Here is another example of that same sort of pathway of the efferent signaling, the central processing, and the afferent signaling. And I'm just showing you what I am expecting you to walk me through on this final exam. This here is no different than this. It's just showing it in a different way. So what I'm truly testing you on this time, truly, is can you look at a figure? And can you, let me see if I can pull this back for a second. 1 second here. Can you also read what is happening in this figure and explain it to me successfully. That is what you are being tested on. All right. Now, whether or not you're going to do it with vitamin deficiencies or whether or not you're going to do it with type 2 diabetes, that is entirely up to you. Now, if you want to do type 2 diabetes and you don't know what some mechanisms are, here are some different mechanisms, right? There's leptin signaling, there's lack of exercise, excessive amount of fat cells and overfeeding. This leads to dyslipidemia. So you could look up dyslipidemia. You could look at changes in endocrine system. Like this is that neurohormone thing that we were just talking about. There's insulin. There's microorganisms such as bacteria and viruses. Bacteria would be, this could also be changes in the microbiota in the gut. Anything you want, hereditary diseases, anything you want, you can start talking about, okay? And of course, here are the consequences of diabetes. This would be something you would have to talk about as well. So not only do you have to explain the changes in the mechanisms, right? And again, I said, okay, let's say tyrosine 985 was no longer autophosphorylating. And because this isn't phosphorylating, this isn't activating this cascade of proteins, and therefore STAT3 can't translocate into the nucleus, bind to the DNA, and initiate transcription. Okay, so that would be an example of a mechanism. Then you would have to say, as a result of that mechanism changing, we get fatty liver disease, and we get obesity, and we get osteoporosis or whatever. So this is where you have to tell me what changes. And then the final piece will be you having to tell me how exercise, if we increase exercise, how exercise might reverse this, which might reverse these complications here that the body is experiencing, because there are thousands of papers that talk about how exercise can reverse mechanisms, because that's what we do as clinical exercise physiologists. You have to understand the disease. You have to understand the mechanisms to understand how exercise might reverse it. And that's why exercise is medicine. So let's move on past obesity. If you wanted to do cancer, we know that obesity and cancer are directly related. They overlap. Let me see if I can find that mechanism. It's called the MAP kinase mechanism. MAPK mechanism in glucose uptake. So just giving you guys another example here. Let me see if there's a picture. So essentially, yep, here's a simplified one. There are two different pathways. Nope, that's metformin. I don't want to show you that one. There are two different pathways that glucose activates in the cell. One of them is activating glycolysis, and the other one activates the MAP kinase pathway, which is going to tell the cell to grow and divide. So when we have insulin resistance in diabetes, here we go. This is what I was looking for. Let me see if I can make this bigger. Nope. Give me 1 second. Just trying to make this, find the right picture for you guys. So when we have the development of type 2 diabetes and we are resistant to insulin, we still activate another pathway in the cell through insulin, which essentially tells the cell to divide and to grow. Here we go. This is what I was looking for. See if I can make this a little bigger for you guys. Okay. So something you might want to talk about as another mechanism. So I'm just giving you examples of mechanisms. When insulin binds to the insulin receptor, it will activate the glucose uptake pathway, right? And you guys know this, especially from my classes before. This insulin will autophosphorylate this insulin receptor. This tyrosine receptor will phosphorylate IRS1. IRS1 will bind with P85 and P110, which will activate PI3K. PI3K will activate AKT. AKT will be phosphorylated, and AKT will activate mTOR, which you guys know is important for muscle growth. mTOR and AKT will activate this GLUT transporter, and this will let glucose into the cell. This is a mechanism. Now, when we develop type 2 diabetes, we can have resistance here. We can have damage from to any one of these enzymes through free radicals. This is why I taught you about free radicals. These things can be produced and they can shut off anything here because, as you know, they bind to proteins, they bind to lipids, they can bind to anything that has amino acid residues, and they can destroy them. So if we have insulin binding, but we have resistance either at the receptor or we have a lot of ROS being produced and it's shutting off things inside, well, that's going to stop the GLUT transporter from letting glucose into the cell. So now we get high levels of glucose in the blood that can't get into the cell because it's resistant. Now, even though in diabetes, this pathway is shut off, this other pathway is still on, and when insulin binds to it, we activate this MAP kinase pathway, and this MAP kinase pathway tells the cell to grow and to divide and to continue to grow. So one of the first things that happen when we get insulin resistance is that the beta cells produce and secrete more insulin to try to compensate for the glucose not getting in. So even though we have all this extra insulin circulating, glucose isn't getting in, but guess what? It's still activating this other pathway, which is this kind of carcinogenic pathway, which is going to tell the cell to keep growing, keep dividing, even when it's absence of energy. So that is how obesity and cancer are related. There's also IGF is related to cancer. So again, I'm just showing you examples of mechanisms. I'm just trying to show you what I'm expecting from you guys. So if you guys want to talk about insulin resistance, you have to be willing to walk me through this. And any paper that you look at will give you an example of what is happening here, but you need to explain that to me. Okay, and then here is my last kind of example. This is the IGF axis. This would be another example of how diabetes changes. This is IGF, which is insulin growth factor. This is another hormone, and you can talk about how IGF changes with the development of obesity. I'm just giving you guys examples. All right, you make this your own. And then on the last few slides here, if you don't want to do obesity and you want to do vitamins, well, I have some examples here. I'm not going to read these to you. I'm going to let you do this on your own. Here are some examples of deficiencies. I give you some examples of what happens with vitamin deficiencies. So, of course, vitamin A deficiency leads to blind spots, right? Also night blindness when you can't drive at night. But you can see here that I put a mechanism up, and there's changes in cell differentiation when we have vitamin A. And these changes in cell differentiation will change the eye and how the eye works. And we can develop almost like this plaque in vitamin A deficiencies. So let's say you want to talk about that. By all means, talk about it. This plaque also leads to night blindness and blind spots. And you can see I put some rodents up here that have that same sort of plaque from a vitamin A deficiency. And this is from epithelial cell changes. Leads to night blindness. Another one would be vitamin D deficiency. So if you want to tackle vitamin D, this one's super complex, right? The more complex it is, the more points I'm going to give you guys. If you want to walk through vitamin D deficiencies, you want to tell me about how it starts, how it initiates, what mechanisms are involved, and again, how exercise, I'm just going to put EX, either will make it worse or perhaps make it better. So this is for you guys to tackle. If you want to talk about more vitamin D deficiencies, you can talk about how it impacts the kidneys and the liver. So vitamin C as well, here you go. And I put some mechanisms here for you guys. And then if you wanted to talk about, again, for exercise, really important one would be vitamin B complexes. So that is what you guys are going to have to do for me. You guys are going to have to explain what the deficiency is if you talk about vitamins. If you talk about obesity, you have to tell me about what the changes are that are going to occur as a result of obesity. Then you are going to have to find some mechanisms, and it's totally up to you, whatever mechanism you want to talk about. And then I expect you to bring in things from our previous conversations. We've talked about exocytosis, endocytosis. We talked about the different types of cellular receptors. We talked about gene activation. We talked about DNA. We talked about inflammation. We talked about adaptive immune response. We talked about chronic inflammation. We talked about acute inflammation. We talked about neutrophils, macrophages, T cells, natural killer cells, adaptive B cells. We've talked about all of these different things. We've talked about hemodynamics. We've talked about free radicals. I have given you guys as much as I possibly could at the pace that I thought was safe for you guys to go at. Now I need you to put it all together and blow my mind. So I will get you these your directions next week. I just want you to watch this video and be okay with where we're going. And then I'm going to leave you guys alone for the next 2 weeks so you guys can put this together and show me that you are capable of presenting like a master's student who has just taken pathology. That's all I got for you guys. Take care. I will be in touch soon.